All right, welcome back. We're going to now pick up and talk a little bit about the cell-mediated immune response. So we've already covered humoral immunity and how this involved B lymphocytes. They can bring in and process antigens as an antigen-presenting cell. They're then able to proliferate and differentiate into memory B cells and then plasma cells, which secrete the antibodies. Now we'll go and we'll talk a little bit about the cell-mediated immune response, and this is going to involve our T cells, but we'll also introduce a little bit, or we'll talk again about some B cells as well. So, thymocytes is just another name for our T cells because they mature in the thymus. Our thymocytes are going to express some molecules on their surface known as cluster of differentiation. So clusters of differentiation, there's 4 and there's 8. So sometimes these are just abbreviated CD4 markers or CD8 markers. Now, CD4 markers are present on T cells, which will become helper T cells. Sometimes they're denoted T subscript H for helper T cell. Those T cells that contain CD8 markers will become cytotoxic T cells, or T subscript C, so cytotoxic T cells. These perform slightly different functions. Helper T cells are responsible for activating other cells, and then cytotoxic cells, cytotoxic refers to something that is toxic to the cell. These are the ones that actually act to destroy foreign or cancerous cells. Now, these T cells, unlike B cells, the T cells are only able to recognize to antigens that are processed and fragmented and then presented on the surface of an antigen-presenting cell. So you remember we talked about macrophages, B cells, and the dendritic cell. Now, dendritic cells appear to be a very good cell at presenting and stimulating naive T cells that have never been activated before. So the dendritic cells, which is a type of antigen-presenting cell, seem to be very effective in doing that. Now, clonal expansion refers to this idea that our T cells, once activated, can divide and expand into clones of itself. So they can go to, we'll see there are memory T cells and regulatory T cells as well. But it requires recognition of 2 things. You have to recognize a self molecule, so the MHC molecule that we talked about, and then they have to recognize the non-self or the foreign antigen that is being presented on the MHC molecule itself. So you've got to have this dual recognition, and this idea of recognizing something self and recognizing something foreign or non-self helps to reduce the likelihood of an autoimmune response occurring and us attacking our own normal self cells. So we talked about how there were 2 types of MHC molecules, and we introduced one with the B cell. We talked about MHC class 2 that is present on antigen-presenting cells. Now the other one is called the MHC class 1, and these are on all body cells that contain a nucleus, so all nucleated body cells. And so the only cell in our body that does not contain a nucleus is our red blood cell, so you're not going to find MHC1 molecules on red blood cells or the erythrocytes, okay? These MHC1 molecules will present an antigen on the molecule that presents on its plasma membrane, and these MHC1 molecules are going to interact with the CD8 T cells. So if you remember, we said CD8 were the cytotoxic T cell. So there's an easy way to remember which MHC molecule responds to which T cell, and I'll tell you that after we talk about the MHC2 molecule. So the MHC2 molecules are going to be those which are presented on the surface of our antigen-presenting cells, as we mentioned with our humoral immunity part of this lecture. Now, when you bring in a molecule into the antigen-presenting cell, it is fragmented and broken down and then presented on this MHC2 molecule on the plasma membrane of the APC. And then these MHC2 molecules will interact with a CD4 T cell. And if you remember, CD4 T cells are the helper T cells. So, as I alluded to a second ago, there's an easy way to remember which MHC molecule interacts with which CD8 T cell, or which CD T cell. So MHC1 will always respond to CD8, and MHC2 will always respond to CD4. So the easy way to remember this is you have to get to 8. Okay, so 1 MHC1 times 8, CD8, that equals 8. 1 times 8 equals 8. When you go to MHC2 times the 4, CD4, 2 times 4 equals 8. Okay, so this is a really easy way to remember which MHC molecule is going to interact with which T cell. Now, the CD molecule, CD8 and CD4, those are present on the T cells. MHC1 and MHC2 are present on the other types of cells. So MHC2 for the antigen-presenting cells and MHC1 on our other body cells that have a nucleus. So here you can see the interaction between MHC molecules and helper and cytotoxic T cells. So again, we have an MHC1 molecule, so it's going to interact with a CD8 T cell. And we said CD8 T cells are our cytotoxic T cells. So you can see those two interacting here. Now you can see the class 2 MHC molecule, and it interacts with a CD4. So 2 times 4 is 8. And we said our CD4 molecules are going to be present on helper T cells. And so now you can see those interacting. And remember, MHC2 are on our antigen-presenting cells, and that's what we see right here. This is an antigen-presenting cell. So as you see these things, try to think about how all of these things are interacting. So if you see antigen-presenting cell, you should think macrophage, B cell, or dendritic cell, and then always associate which MHC molecule with the corresponding appropriate T cell. Now, once we present the antigen on the surface of the antigen-presenting cell or the other cell, we have to activate our T cells because they have to be activated in order to function and carry out their role. So what you have is you have your MHC1 interacting with your CD8, you have your MHC2 interacting with your CD4, and you have to have co-stimulation. So this is what I was saying, you have to have interaction between the T cell and self-antigens as well as the foreign antigen that's presented on the MHC molecule to prevent an autoimmune reaction of stimulating and attacking our normal healthy cells. So we have that, and after we have our co-stimulation occur, we can have our antigen-presenting cells and our T cells secrete different chemicals, and when these chemicals are secreted, they stimulate the proliferation and differentiation of our different types of T cells into memory T cells and regulatory T cells that will be used for later use. So here you can see this. It looks like a lot, but start at the beginning for our CD4 and our CD8. So we'll start with CD4. So what we see here is we see an antigen-presenting cell because we have a class 2, and you see our CD4 helper T cell. What you see is a T cell receptor, and it binds with the MHC, it binds with the antigen or the epitope presented on the MHC molecule. You can also see the CD4 molecule interacting with the antigen-presenting cell itself. So this is what will happen to prevent the autoimmune reaction. So we make sure we have co-stimulation of both the foreign antigen and a self-antigen along the antigen-presenting cell. Now, once this happens, you can have the helper T cell become activated. Okay, so the helper T cell becomes activated via cytokines from the antigen-presenting cell. These cytokines that are now released from the helper T cell can then go and stimulate the proliferation of the cell into helper T cells or memory T cells. And then it can also stimulate the proliferation of our B cell to actually secrete, to divide into a plasma cell, and then ultimately secrete lots and lots of different types of antibodies. Now, with our cytotoxic T cells, or our CD8 T cells, we see a similar thing, except now we have MHC1 in our infected cell, and we have our CD8 cytotoxic T cell. But again, you see the T cell receptor interacting with the epitope presented on the MHC molecule, as well as the CD8 marker on the T cell itself. But what you see here is now we see something called perforins and granzymes. So what these perforins and granzymes are is when the cytotoxic T cell becomes activated, okay, and it can become activated via our helper T cells up here, it will release perforins and granzymes. So perforins will go to the infected cell and essentially form lots and lots of small little holes in the plasma membrane of the infected cell. So that's what you can see right here. All of these little surface markers here, these are all holes that have been punctured in the infected cell via the perforins. Now, once the perforins are in place and have put in these holes, granzymes can come in, and these are proteins that will act to break down and destroy the infected cell, resulting in it lysing and being completely destroyed. So this is the mechanism, or at least one of the mechanisms of action of how our cytotoxic CD8 T cells will actually destroy foreign or infected cells in our body. So we'll talk a little bit about the specific types of T cells. So we have our helper T cells. Remember, these contain CD4 molecules. They'll interact always with MHC2 molecules, because 2 times 4 is going to equal to 8. They can secrete interleukins, and there are different types of interleukins, but what these do is these stimulate different cells to proliferate and differentiate. The helper T cells will be those that stimulate our cytotoxic T cells to actually divide and become active. And they'll also coordinate activities between different types of cells. So they're kind of an intermediary between some of the B cell and the humoral immunity and some T cells in our cell-mediated immunity. Our cytotoxic T cell are going to be those that actually perform they're the workhorse of the cell-mediated immune system that actually destroy foreign or infected cells. They always will bind with the MHC1 molecule. So if you remember, CD8 and 1, that equals 8. So those two will always go together. They secrete the perforins and granzymes. If you recall, perforins induce the pores in the infected cell, and then the granzymes will come in and actually break down and cause the lysis of the infected cell. And they can also target normal self cells that have now become abnormal, so in the sense of cancer cells. Now, we also have regulatory T cells, and these perform exactly what you would think with the name regulatory. They regulate the activity of B and T cells to release different chemicals and cytokines, and they suppress activity when it becomes too active or too activated in either B cells or T cells. And then our memory cells are those similar to the memory B cells. They're long-lived. They're able to respond to the same antigen that occurs at a point down the road, but they contain the same specificity as their original parent cell. So they'll respond to the same antigen during subsequent exposures. So we've talked about foreign cells, and then we've talked about self cells and all these different things. So this relates very important and very importantly for a clinical implication in the sense of organ transplants. So you can have different types of organ transplants, and we'll go through some of these here. So autografts, auto means the same or self. So this is going to be tissue that is going from one area of your body to another area on your body. So you don't have to worry really about organ rejection in this sense, because it's still your same tissue. Okay. An isograft is that which is donated to another person from somebody who is genetically identical, so an identical twin. We have allografts, which come from another individual of the same species, but it's not somebody who is genetically identical. So this could be this is a common type of tissue or organ transplant. Somebody who is from the same species, so another human, but not necessarily somebody who is genetically identical to you. And then you have a xenograft, which is somebody or a tissue or an organ that is coming from a animal outside of your species, so a different animal species. So this is common with porcine or pig heart valves that can be placed in individuals with a valvular heart disease, or for some other reason they need a heart valve replacement. Now, we have these tissue transplants, and the reason they work or they don't work, and what we mean by working is they are able to take place and hold and be accepted by the recipient's body, or they're not, it's based on the MHC molecules, really. So if you think about what that word MHC means, the abbreviation MHC, histocompatibility. So histo refers to tissue, and compatibility refers to the idea of being compatible. So are these tissues compatible with each other? And so it is best if you have somebody who is an immediate family member donate an organ or tissue, because they are genetically very similar to you, and you want as many of the proteins on the donor tissue to match those as the recipient's tissue, so you have an increased likelihood of the tissue or the organ actually taking place. Now, when this happens, whenever you have an organ transplant, oftentimes these patients will be on something called an immunosuppressant. So an immunosuppressant is essentially a medication that is going to dampen the immune system in order to prevent your body from targeting this newly donated tissue. So you can imagine, if you're taking an immunosuppressant to prevent your immune system from acting and destroying this newly transplanted tissue or organ, what is that going to leave you open for or put you at risk for? So hopefully you're thinking it's going to put you at risk for an infection, because you're not just dampening your immune system targeting that tissue or organ transplant, you're dampening your immune system in general. So this can open up the possibility of infections that you ordinarily wouldn't have to encounter. Now, we'll go over just a few last things here. There are different types of immunodeficiencies that have to do with different types of immune cells and their and essentially them being defective or altered in some way. So the first one we'll talk about is something called severe combined immunodeficiency disorder. So what this is it is a combined disorder of both T cells and B cells. And oftentimes it will result in the individual, the patient, dying from an opportunistic infection. So what an opportunistic infection is an infection via a fungus or a bacteria or a virus, something like that ordinarily would not kill you or cause you severe harm or damage, but an individual who is immunocompromised or immunodeficient, these infections can now become very serious and even deadly. So an opportunistic infection is just something that ordinarily would not cause you many problems in a normal, healthy individual, but in somebody who is immunocompromised, it can be very serious. Now, the next disease is something we've likely all heard of, is acquired immune deficiency syndrome, also known as AIDS. And what AIDS is due to, it is due to the human immunodeficiency virus, or HIV. So, you can have HIV and not have AIDS. Okay, you can have the virus but not have the syndrome. You can have virus and not have AIDS. If you have AIDS, you have to have the virus, because the virus is what causes AIDS. So make sure you understand the relationship between what the virus is and what the syndrome is. And what AIDS actually affects is it affects the CD4 T cells. So if you think about what CD4 T cells are, those are going to be your helper T cells. So if you inhibit and destroy your helper T cells, now you've lost that critical component of your immune system that is going to act to stimulate and activate different other cells in our immune system. So this last part that we'll talk about are hypersensitivity reactions. Okay, so a type 1 hypersensitivity reaction is going to be something that is very immediate, and it occurs due to histamine release generally after an immunoglobulin like IgE binds to something like a mast cell. So this is going to be something like allergies, but it can also be something very serious and critical, like an anaphylactic reaction. So an anaphylactic reaction is a severe allergic response that can be very dangerous and deadly if not quickly corrected. A type 2 hypersensitivity involves IgG antibodies, and this has to do with incompatibility between blood, so blood transfusion reaction. So as a nurse, you will administer blood to patients, and you generally stay in the room. You always type and cross-match to make sure the correct blood is going to the correct individual. But even then, sometimes the patient can still have an immune reaction to the blood transfusion. So you'll always stay in the patient's room for about 15 to 20 minutes after you start the blood to monitor for symptoms of a blood transfusion reaction. And then erythroblastosis fetalis is another type of type 2 hypersensitivity reaction. And what this is this has to do with the incompatibility between mother's blood and the fetus's blood. So between the mother's blood and the fetal blood. And we talked about this a little bit with the blood lecture. But what it is when a mother is Rh negative and the fetus is Rh positive, there can be a mixing of blood sometimes between the two. And what will happen is the mother will develop Rh antibodies that will target the Rh positive antigen on the fetus's blood. And during a subsequent, it's usually not an issue during the first pregnancy, but if there is a subsequent pregnancy in which the mom is Rh negative and the fetus again is Rh positive, you can have this condition known as erythroblastosis fetalis, in which the mother's newly formed Rh antibodies, negative antibodies, will target the blood cells of the fetus and result in the lysis, the hemolysis of the baby's red blood cells. So it can be very, very serious. So there's the RhoGAM injection that is given to mothers to prevent this from happening if they are Rh negative and their fetus is Rh positive. Now, type 3 hypersensitivity reactions have to do with the formation of soluble antigens and antibodies that aggregate in blood vessels and they clump up. So a common one is something called systemic lupus erythematosus, or lupus is sometimes what it's just referred to as. What this is it has to do with these antigen-antibody complexes being deposited in blood vessels, and you can see symptoms related to this. Oftentimes, individuals with lupus, they can have lupus nephritis, which is a common condition in which the kidney is affected, and this is generally a leading cause of death, the nephritis, this renal failure in patients with lupus. And then lastly, type 4 are going to be our delayed reactions, so things like a tuberculin skin test. So if you work in healthcare, you already know you get a tuberculosis test. It's a skin test that goes directly under the skin, and it creates this small little raised portion under the skin, and then it will be read in 48 to 72 hours. And if it is a positive reaction, so indicating that you have been exposed to tuberculosis, the region will be very red and even more raised, and it will be more firm. And so then if this is a positive tuberculin skin test, you generally go and have a chest x-ray done to see if it is active tuberculosis or something called latent tuberculosis. So is it an active infection, or is it something you have in your body but it's not currently active? Is it latent? Okay. So this is just a graph showing these different types. You can have a type 1 immediate, type 2, which would be our blood transfusion issues, type 3 with our lupus, and then type 4 here with a tuberculin skin test. So this concludes our lecture on the immune system. So there's a lot of information with this immune system. So go back to your notes, watch this video multiple times, and as always, email me if you have any questions. I'll see you in the next video.